This is an instructional video on how to administer clinical research medications via IV infusion. Why is the process for giving clinical research medications different from our usual method? Rationale: we must ensure that 100% of the drug is delivered to the patient in the time frame ordered. These standards are dictated by the Clinical Research Study Protocol and FDA regulations. Clinical trials determine the efficacy and safety of new medications, so we must ensure accuracy. Did you know when you leave medication in the reservoir chamber and tubing, you are not delivering approximately 30 milliliters to your patient? So if you have a 100 milliliter bag, that means 30% of the medication was not given. That is a significant dosing difference from the order. Study medications will come from investigational pharmacy Monday through Friday, 8 a.m. to 4:30 p.m. CCH9 satellite pharmacy will handle study medications during off hours, including weekends. All study medications will have a medication label with patient information, medication name, dose, and infusion time frame. They also have a barcode for BCMA. Important: all clinical research study medications are run as a primary infusion. Rationale: exact start time of medication is a critical data point in clinical research. The supplies you will need is the clinical research study medication, primary pump tubing set with injection port above the pump, 50 milliliter normal saline bag for flush, secondary tubing set, and appropriate PPE. The PPE need will be determined based on the patient's condition and the type of study medication being administered. All clinical research medication require 2 licensed providers to verify the medication order for accuracy. This should be done at the bedside whenever possible. However, if a person is on isolation or contact, it can be done outside of the patient's room. Here at VCU Health, our patient identifiers are name and date of birth. Since all clinical research medications have barcodes, we should use our BCMA process to ensure that we have the right patient and order for the medication. Since all clinical research medications are treated as primary infusions, you will spike your bag and prime your tubing like you would with any other medication, but it is essential that you do not waste any medication and that you stop the priming just prior to the end of the tubing. It is okay if there is a small amount of air in the tip so that you do not waste any of the medication. You will program your pump like you do for any other medication, and you will go under Guardrail IV Fluids and select Investigational Med, because it is essential that you use your Guardrail library. If for some reason you do not have Investigational Med as an option, then it is acceptable to program under Basic Infusion. Your infusion rate will be part of your medication order. The volume to be infused should be the amount of fluid in the bag less 25 milliliters to account for priming of the tubing. Please note that the clinical research medication IV bags may be under or overfilled, so when the 75 milliliters is complete, you may still have a little drug remaining in the bag, or there may be some in the reservoir chamber. This is perfectly acceptable. After priming your normal saline tubing for your flush, you will attach it to the Y site of the primary tubing just above the pump, ensuring that the roller clamp is closed. Now that you have attached your normal saline flush line, you will program volume to be infused for another 10 mLs to empty the medication from the reservoir chamber and tubing. You must stand and observe as the medication empties from the reservoir chamber and tubing to the Y site where your normal saline flush is attached to avoid getting air in your line. Please note that if your bag is overfilled, you may need to program volume to be infused for 10 mLs a few times for the medication to empty into the Y site. Note how nurse stands and watches as the medication empties the tubing and ends right above the Y site where the normal saline is connected, and then pauses the pump. The nurse will now open the roller clamp to the normal saline flush and program the pump volume to be infused for 30 milliliters, and this will ensure that 100% of the medication has been delivered. Please note that the rate remains the same as the primary infusion because you are still infusing the medication at the ordered rate. You will note here after the 30 milliliter flush that the tubing is clear, which means that 100% of the medication was delivered. When the 30 ml flush is complete, this is your end time for the infusion. This is the time you will document in your clinical note and is a critical data point for clinical research. Unless otherwise specified by investigational pharmacy or the clinical research team, study medication will be disposed of like any other medication. If you would like to save and reuse the IV tubing, you can label your IV tubing per unit protocol. When you spike your next bag of study medication, you will prime the line completely with the study medication as you did with the previous bag. Since you emptied the bag and the line to the Y site, you will be able to track the med in the line as it travels down because of the air in the line. Reminder, do not waste any study medication. You will prime to the end of the line, leaving a small amount of air in the tip.